Good afternoon and good evening, everyone, and welcome back to another edition of Jax Live. Thanks for joining us today as we continue our discussion last week from Model Generation Part 2, Building Complex Models and Embracing Genetic Diversity. I'm Don Tanamachi, Senior Services Program Manager focused on model generation at the Jackson Laboratory. Again with us is Dr. Rob Taft, my fellow Senior Services Program Manager, also focused on model generation, as well as breeding and reproductive services and other innovative technology development. And Bill Buas, Associate Director of Genetic Engineering Technologies team, leading the group of highly skilled genomic architects that can help you design and then bring that custom model design to fruition. As a reminder to our audience, please be sure to use the comment box to submit your questions, add a comment, or just give us a shout out from wherever you're watching. We will address as many questions or comments as possible at the end of our discussion. So as a brief recap, last week we discussed the various types of genetic modifications that are commonly asked for, such as constitutive knockouts, SNPs, or small genomic alterations, as well as the larger modifications as genetic humanization of a gene, adding protein tags, or tracing reporters to gain a function, overexpression, or regulation of specific gene alleles in their endogenous locus or in a safe harbor locus like ROSA26. We also talked about the talented group of study directors that are available to help you distill and design the model, the mouse model, drawing from their experience managing and overseeing hundreds or to thousands of genetic modifications and mouse model projects. We ended up how we validate what we've created, that we've created the desired genetic modification. So in essence, Jax can go from basic design to delivering an actual mouse model and beyond, as we can provide you breeding services and perform in vivo analysis services on your new custom model. And while we touched a little on these individual building blocks to generate more complex models, I really wanted to dive in deeper with this today. So one modification that we talked about is the constitutive knockout. However, sometimes biology gets in the way, and studying a critical gene function using a straight knockout is not always optimal or even achievable for a variety of reasons. Or if we want to regulate turning on or even turning off or even on a gene expression, we can make a conditional knockout or knock-in by introducing LOX-P sites or a lock-stop LOX cassette in certain regions of the genes. So welcome, Rob. Thank you for being here. And what exactly are these recombinational components, and what are the critical things to think about when creating a conditional allele?
Thanks, Don. It's good to be back with you and Bill again this week. As you mentioned, we're going to talk a bit more about complexity this week, and the conditional knockouts are definitely more complex, but they have the advantage of giving us much more precise control over gene expression. And so the challenge that comes with that is that we are going to be dealing with multiple alleles, so that's going to add quite a bit of complexity. So in the traditional Kree lock system that we'd use for making conditional knockouts, you're going to have the gene of interest flanked by LOX-P sites, and then in the same cell type, you also need to have the expression of Kree recombinase in order to get recombination events to occur. And so that gives us the ability to have a lot of spatial and temporal control, especially when we begin to consider not just the traditional LOX-P and Kree, but all the sorts of variants that have come along in the last few years. So we have variations in LOX-P sites, we have variations in Kree recombinase, and then people have gotten creative and done things like make Kree inducible. So with the Kree ERT2 system, for example, that system is inducible through administration of tamoxifen. And so that way you can have not just spatial control by having Kree controlled by a specific promoter, but you can have temporal control because now you can add tamoxifen in at a specific time point so that you can have a particular gene expressed or not expressed at a time of interest as well as a place of interest. The challenge in all this is that we now have multiple alleles, so the breeding is more complex. So we're going to have to typically generate a model that has the floxed allele, that is the allele of interest flanked by LOX-P sites, and we're going to generate a model that's expressing the Kree recombinase. Then we're going to have to cross them together. So that adds quite a bit of time and makes for more complex breeding and genotyping. But part of the power in this is you can begin to do some of these things in parallel. So for example, if you were interested in the impact of the expression of a particular gene in 3 different tissues or 3 different cell types, you might be able to cross the particular floxed line with 3 different Kree driver lines. And so rather than doing these things sequentially, you can be doing them all in parallel, and you'd have much more precision than you would have if you were just using a constitutive knockout. And people have taken this a step further as well. In addition to LOX-P, we now have things like the FRT flip system, so that you, in addition to LOX-P, you can add other conditions. So you can almost begin to think of this as sort of building logic circuits in which you can have multiple conditions. So if this condition and that condition are met, then you would have expression or not have expression. So this does really get complex, so that the control here you get is absolutely exquisite, but the breeding and genotyping can get complex. To help make some of this easier, we have been collecting Kree strains for many years. And when I looked this morning, I think we had 795 different Kree lines. And you can find all of these through the Kree portal on our website. You can also filter for them. If you're doing a search in Jack's mice, you can just click on that particular filter and filter down to all of the Kree lines to find the one that you want. The advantage of some of the ones that you'll find in the Kree portal is that thanks to support from NIH, there's been additional characterization to make sure that the Kree recombinase is functioning in the way that we think it should be.
Don, on the Kree portal, I can't emphasize enough how valuable that is to this sort of two-part problem that Rob and you guys have just been chatting about. A lot of publications that come out on using the Kree lock system that other investigators might reference and design their experiments around. Sometimes those Kree recombinases work in tissues that were not documented or were not even studied in those original primary literature. And this Kree portal a lot of times has looked at other tissues that weren't part of those primary studies. And so it's almost like someone else has done your experiment for you, sort of your quality control. So it's a great resource.
Yeah. So you can find out, especially if you don't know about certain cell types, you can find out if there a Kree line made in that cell type. I also wanted to point out that in addition to the Kree portal, we have some basic information on the Kree lock system, just to kind of as a refresher, because it does get to be very complex. One aspect that I wanted to bring up is also knowing the background of the Kree lines, especially if certain backgrounds or strain backgrounds that you're required to work on that is not a C57 Block 6 mouse. So, so Rob, I wanted to point this to you also as another example of multigenic systems and moving beyond this traditional Block 6 genetic background.
Sure. So that's something we're seeing more interest in, is moving beyond just Black 6J. And there are a couple of things I was thinking about when you just made that comment, because this has come up in a lot of projects where someone is looking for the right Kree line. So there's the issue of the background strain. So for example, if you're generating a model and say an NOD background and you made a conditional knockout, you have to make sure you have the corresponding Kree line. And most models at this point are made on a Black 6 background. So it may be hard to find those, and you may have to, the traditional approach at least would be to use breeding and say a speed congenic project to move these from one background to another. With all of the technologies we talked about last week, obviously we can make these models directly. But the other thing I wanted to point out for folks who are looking at things like the Kree portal is to also pay attention to things like substrain. This will come up where someone will look in the portal and they'll see it's Black 6 and they'll see that the Kree that they're interested in is there, but it may not be on the right strain or even on the right substrain. So those are things to pay attention to when you're looking at this.
Yeah, I pretended to be an immunologist in the background, so that substrain difference is very different because people would be B6, I might say B6J, B6N, B6 something else. Sorry, Bill, go ahead.
I think, we talk about this complexity, and there's value to it, embracing complexity around the Kree lock system, and it really brings up this issue of genetic background. A lot of the tools have really been built or bred into the C57 Black 6 genome background, and there's a lot of reasons for that. That's probably another show that we should do. But definitely, there's been years and years ago, mouse geneticists established other inbred strains of mice that have different genetics, different genetic diversity, and that genetic diversity drives different phenotypic diversity. So Rob mentioned the NOD, the non-obese diabetic strain, which was derived in the process in the 60s and the 70s, ultimately published in the 80s. Jack's researchers through the years have embraced the use of that in diabetes research, NOD strains, and then derived from that other strains, which, for instance, breeding other mutations called like the SCIB mutation and the interleukin-2 receptor gamma to produce the MSG strain. Those non-Black6 derived strains have become really important workhorses. And I think it's an issue, or it's an important issue for people to start to explore, and people are embracing, not just the JAKs, but beyond, is these other base inbred backgrounds. The challenge is sometimes if you go in the Kree lock system, a lot of times it's already in the Black6, and so you're partially constrained. But as you know, my group, which has been making mouse models with JAKs researchers pushing these other strains, desiring their research program to use those base strains more effectively, like NOD, we've been doing gene editing for years in the NOD strain and NSG as well, and embracing that level of genetic complexity, the genetic background, as well as the other genes, has led us to what we call heavy use strains. And that heavy use enables us to get a lot of experience because they behave slightly differently than the Black6 strains that many other organizations are used to. And it's really become routine for us. And I think that is really helping us support JAKs research as well as other researchers trying to explore, move beyond the Black6 genome, which is important. But I think there's this extra feature that we really need to embrace.
So something that both of you on too is using it as a routine strain background. And can we use the Kree lock system on the NOD NSGs? Like, can we actually create the Kree driver strains on these one also too?
Yeah, so if you go onto the Kree portal or in JAKs, a lot of those Kree strains are on Black6, which might pose a problem where someone's like, well, I don't have my Kree strain on NOD, but we are effectively using this strain regularly, and we're making Kree-based strains on NOD as well as NSG, as well as the Flox alleles on a regular basis. It's actually very efficient.
Great. So sorry about the feedback. Important about these multi-gene locus, and then kind of actually, so you mentioned like layering. What is this layering? Or maybe we've been doing this all along.
Yeah, I think we've been doing layering for years and years. So we've been talking about like Kree locks as we started out about two components, two genes, and there's complexity there in breeding. I think the modern gene editing tools have allowed us to start to explore this idea that you can avoid some of those breeding problems when there's multiple genes and they're all segregating and becoming challenging. And it's doing gene editing right on the strain that you want. And I call that layering. So you start with your base strain like NOD, and then you do your gene editing on top of that, sort of avoiding this other many years of breeding problem.
So, so in your work, do you have a good example of who's actually using layering effectively at JAKs?
Yeah, my current favorite case study for layering is this group at JAKs as part of the NIH-funded, it's called Model AD, to tackle Alzheimer's disease, build better mouse models and preclinical animal models. They're one of 5 funded efforts of this consortium. They're doing this layering where on the Black6 genome, they have 2 genes that are known to be sensitizers for Alzheimer's disease in the mouse. So Black6 on its own isn't so great for preclinical modeling, but these other 2 mutations sort of edge it towards. Now they have all these human potential pathological mutations, SNPs, that they're having us mutate on their custom-built strain, which already has 2 mutations, layering those triple mutants on there. And in a year, they have phenotype data on that. And now they're coming back to us with that triple mutant and saying, this is the triple mutant out of all these ones that you made that we want to layer on another one. And it's really these research programs that are helping us iterate. And it's this layering process that avoids all the breeding problems that we talked about.
So that's really cool. Isn't everyone doing this?
Yeah, there's always the rub. And I think we're in the early days of layering 2.0. And as Rob likes to remind me, it takes mice to make mice. And so in order for us to do layering and do gene editing, we need to get enough animals to produce enough embryos to carry out the gene editing process. And there's some gene editing processes that are much more efficient than other ones. So that's one small constraint.
So I keep happening back to my my earlier days of mouse colony management, where we actually had a strain that had 5 transgenes and 2 knockouts. So I was actually doing layering, but I was doing it the very old school way, but requiring, for the knockouts, you need to be a homozygous, you need to have that in a fixed state. And then researchers want to say, yeah, we want to add a different humanization. So it'd be another knockout and another humanized gene. So it was.
Yeah, today we should have gotten together years ago, because then we would have done layering in this process.
But one of the issues that we actually had, because this model had been mucked around with so much, is that it wasn't particularly hardy. So I know this is something else that kind of comes up, and in terms of reproductive fitness, and this is really Rob's area of expertise.
So that is something that comes up, and Bill's talked about sort of our workhorse strains and mentioned Black 6 and NOD, NSG. There are a number of others, but when we move beyond some of those, the challenge really becomes the reproductive fitness of the line, and that is how well does it breed, how efficiently can we produce embryos, and how robust are those embryos. In some cases, after microinjection, the embryos are quite fragile and will have relatively poor success. Others do quite well. So you really have to look at all of those factors. It really impacts the efficiency and ultimately the cost as we sort of move out and branch out into some of these other strains. And what we're looking at in some ways is sort of a negative. We'll also find strains that are better. So there's a full spectrum here when you look at the reproductive fitness. While most of what we use is Black 6, it's sort of actually in the middle. There are some strains that are much better, and there are some strains that are much worse. And so, but that's something that we can help folks through if they're interested in branching out and moving beyond Black 6.
Yeah, and while this Rob described is a constraint, it's a practical thing right now. I think innovation will start to erode that, and that's a good thing to look forward to. It'll allow people to move beyond CPT7 Black 6 as a genome, which is useful. But again, I think there's these other inbred strains and sort of embracing genetic diversity to make the mouse sort of what I like to call is rebooting the mouse to be a better preclinical animal model.
No, very, very much. We don't live in a B6 world all the time, as we are not all the same between us. I did actually want to address one question about what is the advantage of using the Kree lock system. So I think we went through this. There are definitely a lot of websites there that can help you actually go there, which describes the Kree lock system. And as Rob actually talked about, you can actually really impose a really fine-tuned temporal and tissue-specific expression where you can get recombination, so you can actually turn on or turn off a gene. There's also different other pre-locks or pre-locks type systems like the FRT flip-A's system. And I said, really getting that Kree portal again. I'm just going to do a shout out for that Kree portal because it really has a lot of information about existing strains that you can really dive deep, and you don't actually have to make all these models again. The resources there, so this gives you a lot of tools where you can actually build something very, very fine-tuned. I've also even seen lines where they've done an intersectional. So, and as Rob said, we'll talk about this kind of computer program or coding in a mouse where you can actually satisfy two conditions. So it's just another way that you can actually really fine-tune and pinpoint exactly where you want or do not want the gene expressed in that aspect. So any final thoughts on complex models and diversity? I'll pitch it to you, Bill.
For me, it's these new, this layering thing that I like to go on is layering 2.0, and I'm looking forward to being part of layering 3.0, which is helping embrace beyond Black 6, because I sincerely believe that there's lots of genetic variation in the mouse population that's going to help us model human disease that has been known for years. We just all have to kind of rediscover it and not be intimidated by some of these barriers that we've had in place for years.
And last, Rob, any final comments or thoughts?
A couple of real things. Going back to the Kree locks question, you mentioned a couple of resources, but our Technical Information Sciences group, they have a lot of good materials. They have some webinars that I think are available pretty much all the time that would help understand the system, but I think also help understand the breeding and how to breed efficiently when you're using the Kree lock system. I know from talking to the TIS folks, that's something that comes up a lot, is how do you manage the breeding? So that's something we can help you through. And if you still have questions, just call us up and ask. I think Bill covered diversity pretty well, but so due to my role in the cryopreservation program here, I also have insight into some of the things we have. And so we have over 100 different inbred strains that are either alive or cryopreserved. So there's actually a tremendous amount of diversity. All those lines have unique characteristics. And we've really only, I think, begun to explore the power of the mouse as a model system if we consider all of the diversity that exists out there today.
Well, this has been a great discussion. However, unfortunately, we're running out of time. I really want to thank our guests, Drs. Bill Buas and Rob Taft, for graciously sharing your time and all the really helpful information. To everyone out there, thank you for joining us last week and this week. If we didn't get to or answer your question, we'll be sure to follow up with you. If you're interested in having us generate a custom model for your research, please reach out to us at micetech@jax.org or use the link in our model generation site, which you can also find the model generation web form request. I also wanted to let folks know about some upcoming events. Tomorrow, there's a webinar titled Generating Better Mouse Models for Your Research Approaches and Considerations. This is hosted by our TAS group, Peter Kemmelson, and which is our technical information scientist. So next week on Mice Tech Talk, I'll also be walking through that model generation web form and review the information that you'll need when you're requesting a custom model generation project or actually want to talk to a model generation specialist. Lastly, please don't forget to tune into JAX Live next week on Wednesday, September 16th, same mouse time, with Drs. Michael Ellis and Danielle Fontaine as they discuss innovation when it comes to generating new mouse models, diversity, and humanized mice. Thanks for joining.